That's the one I wanted. Really that up there. That's a table on the front.
Bro, do the one that's smaller with you.
That's what I'm looking for.
That water, no shade.
What are you looking for?
No, that water, big water, and shade. Yep. Thank you.
Bingo.
Hey, scholars, Mickey's going to give you time signals of the number of minutes left, right? And it'll be out of 12. So at 7 minutes, 5 left, right? And so on.
Good afternoon, everybody. Thank you all so much for being here today. I think I know just about everyone in here, but I'm Erica Zavaleta and co-direct the Doris Duke Scholars Program with Dr. Justin Cummings, who's also here and close us out at 3 today. I just want to say a little bit about the program before we get started. So today is the conclusion of an 8-week, pretty intense summer that our cohort of scholars, of 29 scholars, have sort of journeyed together. And they are headed back to their homes all over the country in the morning for some of them. So the way that this program is really structured is that this summer is the beginning of a really, really long process that formally unfolds for 2 years, that reality unfolds for a lot longer than that. I also want to acknowledge Bruce, who's here from the Environmental Leadership Program, who's part of the team who runs the formal alumni network for the program. But writ large, I mean, the big goals of the Doris Duke Scholars Program, I'm just going to sort of put a personal spin on this, are really 2 things. One is this very outward-facing goal about changing the world changing the face of his species, changing the way that conservation gets done, and whether it can rise to meet that it faces in the coming decades. So this is the group of people who we've brought together this year. Our growing movement of young leaders represent much more of the perspective of the issue. Should be part of conservation. So, part of what this program is about. Then part of what this program is about is very individual. Right? About connecting people to their passions and helping them find their full potential and find them to do, enabling them to do meaningful work. So, what we're doing today is the first summer of this program is very focused on developing communication and leadership skills, and also on getting an introduction to the process of science and how it works in conservation. This is a very particular lens that we are able to focus on in just 8 short weeks. It's very ecological. It's very field-based, and that has a lot of reasons to it beyond just the science. It's also this opportunity to explore together, to learn about issues firsthand, and then just to kind of have a lot of shared memory to take away from the summer with you together. But this introduction to science piece, I guess what I want to say, for us is really about leadership in science. It's about not just helping other people with their research, but about owning it. And so about formulating the questions and figuring out how to address them through studies that this group of scholars has designed in teams, and learning how to work with the data themselves, and then communicating the findings, like through interpretation, through scoring them with other knowledge in the literature and so on, and then giving talks and writing papers. So that whole start to finish process. And we do that 3 times over the course of the summer. So what that means is that what you're seeing today is just the third of 3 really short opportunities to try that, to scaffold that process. It's not a summer-long research project. It's something more like contained, but also I think what you're seeing is a culmination of the skills that everybody has developed over the summer, right? Because they've tried this now a few times. And some of the students who came into this program this year have done a lot of research in one way or another in their first year or 2 of college. Others didn't have any experience with this kind of thing prior. So, it's a real range, but I think what you'll see today is wherever they started coming into the program, they are now going away with a sense of what it means to be a scientist, to engage with conservation through the lens of research, and then to talk about it, to have that outward face, and to present to all of you who aren't part of the cohort what they did in the White Mountains this summer. So I want to pass it off to Abe, who's going to MC the rest of this morning, and just to thank everybody for coming, and to thank those of you who've been with us for the journey, for an amazing summer.
So I'm only going to MC the first half, because it's been my honor this summer to teach with Tina Chang over here in the audience, and she'll MC the second half. So Tina and I have spent, 6 weeks seeing our stuff go from maybe designing their first experiments, where they really got to own the whole part of the process, to these final projects in the White Mountains. Watching them all grow as leaders and scientists over the summer has been really humbling and fantastic experience. But instead of rambling on, as I could, and gush about how amazing they all are, I'm just going to introduce the first group here. So, our first talk is titled, Dolomite Induces Higher Reproductive Output in Hoary Aster, but Not Sagebrush. And you have a laser pointer, if you would like, and a slide.
Hi, welcome everyone. Thank you all for coming to our presentation. My name is Joe Hernandez.
I'm Karen Monley.
And I'm Rita.
And today we'll be discussing about how dolomite induces higher reproductive output in hoary asters put on sagebrush in California's White Mountains.
So abiotic factors are a really critical driver in biological communities.
And some of those physical characteristics of an ecosystem can include elevation, temperature, humidity and water availability, and also weather patterns. And in alpine systems specifically, some of the physical constraints on diversity are high elevation, low soil temperature, low humidity, and extreme weather.
So for our experiment, we decided to focus on soil substrate and specifically, we decided to focus on dolomite because in the White Mountains, there's two dominant parent materials, and we have the sandstone and quartzite parent material and the dolomite parent material. However, we focus on dolomite because it has three specific characteristics that can largely limit how much growth a plant can have in the area. So the first one is nutrient availability. Dolomite in particular has a really low level of phosphorus, which can make it hard for plants to grow in their early stages. It also has a specific physical composition where it's a sedimentary rock, so when it gets weathered, it actually breaks up into very small fragments. And this can affect like the water availability for the plant, the aeration it's receiving, as well as the resistance to root penetration that it can get. And finally, it also has a poor microbial community, and this can largely hinder like general plant.
Okay, so as mentioned before, our studied area was the subalpine of California's White Mountains, which has the soil gradient from quartzite to dolomite, as Maria just mentioned. And you can see that transition of soil in this picture, quartzite being on the left and dolomite being on the right. And being on site, we observed that there was a higher population of sagebrush, Artemisia trinitata, or sagebrush, which is, the plant on the left. But as we went up higher up the gradient towards the dolomite substrate, we saw there was less of the sagebrush and more of these small flowering plants. One that stood out to us was the Diatericanthus, or the Oriasters, which is those purple flowers on the right. And so we decided to investigate how this transition of soil gradient from
This is the wrong presentation. I'm sorry.
Just keep going, yeah.
You guys don't want the other one to be posted? Sorry, guys.
Right, so we decided to choose these two specific plant species because they represent two very different life history strategies, considering that the sagebrush is pre-annual and the asters are biannual. So we decided to see how this dolomite gradient impacted these two very different plant species.
Yeah, and what?
So the way we phrased this, sorry guys, the way we phrased this question into our question was that how does the reflective effort of sagebrush and asters differ across substrate gradient across the dolomite? And the way we wanted to measure reflective effort was through flower density, which is the number of flowers per plant area. And so the way we expected to see this was that across the dolomite gradient, we will see a higher flower density increase for the asters, but simultaneously we will also see a decrease in the sagebrush flower density across the dolomite gradient, because this kind of reflects how we expected very different reactions between the two plant species due to their different life history strategies.
Yeah, so one of the things that we're trying to get at with our question is that depending on the life history strategy of the species, they're going to have to make different trade-offs. And a lot of times when environments are stressful, the first thing to go is a reproductive output, and plants will instead focus on things like growth and survival. So we're trying to see what that sort of optimal region is during which reproductive output will be the highest.
Our main methods to study our question was setting down a 100-meter transect line where for every 20 meters we did a 1 by 2 meter plot. And in this plot, we counted the number of individual plants as well as the number of flowers on each individual. We also counted the area for each plant using the length and the height. And as you can see in this picture, we also did a percentage of dolomite coverage per site.
So when we did this, we were able to see how increasing dolomite changes flower density. And so if you look at our x-axis, we have dolomite percentage, and on the y-axis we have mean flower density, which we measured as number of flowers per centimeter squared. And If you look at the solid line, which represents our asters, you can see that as the dolomite percentage increases, so does the mean flower density of flowers for asters. And one of the reasons for why we think this could be is because if you think of the dolomite gradient as a stress gradient, where higher dolomite means a more stressful environment, it makes sense that given the really short lifespan that asters have, if they're in a really stressful situation, it doesn't really make sense for them to conserve a bunch of energy for growth and survival, since they're probably not going to survive until the next reproductive season anyway. So in that really stressful situation, you have really high reproductive output. Whereas for the sagebrush, which is a much longer-lived plant, it can live over 50 years. Some years might be more dedicated to reproduction, some years might be more dedicated to growth and survival, but you don't really get that same trend as you do with the asters.
Okay, so when we looked at the plant area for each individual plant species, we noticed that there was a trade-off in the asters life history strategy. So on the left, you have the graph for the asters and mean plant area. So the y-axis is our mean plant area in squared, and the dolomite gradient being in the x-axis. So you can evidently see that there's a decline in the asters plant area, and this decline in the plant area could be due to the asters strategy to its trade-off in trying to trade off in plant growth and plant area for its reproductive effort, which Huey just mentioned in the past slide. And we see this similar suggested pattern for the Artemisia in the right graph. So on the y-axis, we again have the mean plant area in meters squared and dolomite on the x-axis. And we also see this decline in the plant areas across the dolomite gradient. But as Kira just mentioned in the previous graph, there is no increase in the effort for its reproduction. So this decrease in plant area could be due to the lack of core nutrients that dolomite failed to provide, and in doing so, it induced stress on the sagebrush, so it limited its plant growth and plant size.
As our final measure of reproductive effort for sagebrush and for aster, we decided to look at the coefficient variance, just to see if, like, on average, like, the variability across sites for each plant actually differed. And what we noticed was that although across the dolomite gradient, there actually wasn't as much variance as we expected, on, like, relative to the mean, sagebrush actually differed more than aster, which makes sense given its, like, longer lifespan. And since it lives for a longer period of time, it can have more reproductive years or less reproductive years, giving it more of, like, which may cause it to have more variance. While the aster has a very short lifespan, which suggests that in general, it should have a similar reproductive effort throughout its lifespan.
Yeah, so now if you look back at the first picture that you showed, you can really see that with the change in soil, you also get a change in vegetation. And understanding how soil can act as a driver for vegetation changes is really important because plants are the primary producers of any ecosystem, and a lot of other trophic level diversity depends on the vegetation of an area. So understanding how soil can be that driver can help us understand the interactions between other trophic level diversity, and also help us predict how ecological communities will actually respond to environmental changes.
So we'd like to thank the Doris Duke Foundation for funding our research project. We'd like to thank the Doris Duke Conservation Scholars Program staff for helping us in every step of the way. We'd like to thank all the scholars for helping us with more support throughout the whole process. We'd like to thank you guys for listening to us. Any questions?
Yeah, so I'm curious how you figured out what the timing was for flowering of the key study species that you focused on. It's a super interesting thing to compare these life history strategies, but I'm also wondering how much of the variation you might have seen might have been due to timing differences and when they have flowers.
Yeah, so one thing that we also measured that we didn't mention was the number of buds on all the Artemisia. And there were a lot of them, so we actually could only look at a single stem, count how many were on that stem, count the number of stems. And for some of the individual plants, we got like over 1,000 buds. So we assumed that that meant that not every single bud would become a flower, and it definitely would have been interesting to delve into that more. We did see similar results, the same patterns between number of flowers on the sagebrush and number of buds. But yeah, I mean, we don't really know what percentage of those buds would turn into flowers within like the next month.
That is why we focus on reproductive effort. So by doing the bud thing, it just helped us out. So instead of measuring like how much actual flower output there would be, it was easier to just measure how much they're trying to make, you know? So.
A question about since you're interested in the sort of micro patterns of these different species. When you look at the sagebrush, did you see a lot of small ones too? Like, were you able to figure out anything about like what the cast might look like for those populations? Do you think they just have one really big year and all of a sudden a lot germinated and they all kind of are the same age? Or do you see
We did see a higher, like larger sagebrushes at the bottom of like the gradient of dolomite, but we should look into more research about like how at what age stage each sagebrush was across the substrate.
Yeah, and for sagebrush specifically, size isn't really a good measure of age. There's other ways to look at age for that, but that's not something we look at in ours.
Different patterns on the other side of the gradient on the same foot track, and you notice that those fishes are different or they're flowers.
Well, physically speaking, that's actually why we did the question, because when you look at it, the sandstone quartzite side has like a whole bunch of sagebrush and it's like bigger and seems more fruitful until we did the actual study. But as you go towards the dolomite gradient, that's when you start seeing like more sparsely distributed sagebrush, and that's when like the little asparaceae type plants start to show up a lot. Any other questions? Thank you.
So much. All right. We are going to keep the sagebrush theme going here and welcome Julie, Siani, Serena, and Carl. Tell us about the physical effects of the likelihood of galls on sagebrush.
Yeah, I'm going to give you time signals. 5, 3, 2.
Welcome and thank you for attending our presentation. My name is Serena Barassi.
My name is Tiani Batten.
My name is Carl Brown.
And today, we're looking at the physical and biological effects of the likelihood of galls on sagebrush. Keystone species have a large ecological influence on other species in their ecosystem. As a dominant vegetation in the western United States, sagebrush often serves many functions, such as a resource for habitat and nutrient for other species. Thus, plants and animals alike rely on sagebrush as a means of survival. Gull insects in particular rely on sagebrush for reproduction by creating abnormal growths on sagebrush in which they lay eggs. Understanding the physical and biological factors that drive this host-parasite interaction is important for maintaining the gull insect communities in this ecosystem. This information could potentially map out the distribution of gull insects. The biological importance of this is that insects are not only contribute to the biodiversity, but they also are important indications of biodiversity in certain ecosystems. The particular host-parasite interaction that we're looking at today are the insects that form the sponge gallmage and the woolly bud gallmage on big sagebrush. However, past studies haven't really focused on the subalpine environment, which we were interested in. Thus, we asked the question, what physical and biological factors contribute to the likelihood of galls on sagebrush? And we looked at soil moisture, surrounding sagebrush, diameter, and whether it was located on a sloped or flat land. And we also looked at whether the sagebrush was actually infected and how infected it was. After initial observations that there were remarkably less galls in the wet areas of the subalpine environment, we came up with two hypotheses. First, galls are more likely to be present in sagebrush in the drier areas, and second, galls are more likely to be present on sagebrush on slopes.
So in order to test our hypothesis, we wanted to look at Campedo's meadow and use that as our sample site. We chose Campedo's meadow because it had the wet to dry gradient that we wanted to use, as well as the slope versus flat land. So this pictured here is Campedo's meadow. And as you can see, in the center of the meadow, the land is flat, but the further out you go, the land becomes steeper and there are slopes. As Serena mentioned previously, we wanted to look at soil moisture, whether or not there was a slope, sagebrush diameter, and percent coverage of other sagebrush. We tested for the percent cover by looking at all the surrounding sagebrush within 1 meter of our focal sagebrush. We took the sagebrush diameter by calculating the maximum diameter in meters. And when it comes to the presence or absence of galls, we would physically manipulate each sagebrush by moving around the branches and the leaves and asking ourselves one question. Are there galls on this sagebrush? And if so, how many? Before moving on to our next sample, we would collect soil that was located directly next to our chosen sagebrush and take it back to our lab. That same day, we would weigh the soil, and then we would let it dry for a few days, and then we'd weigh it again. We did this to get the percent of water that was in the soil, meaning we took the initial weight and subtracted that from the final weight, and then divided that by the initial weight.
So here we have a figure showing the gall presence on individual sagebrush on the y-axis and the percent water found in the soil on the x-axis. The data points located at 1 represent individual sagebrush that had a gall presence, and the data points at 0 were the individual sagebrush that had no gall presence. So now this is the same exact graph with the best fitting curve, and so we found that there was a strong relationship between gall presence and the percent water found in the soil. And as you can see from looking at the figure, the gall presence was more likely in areas where the percent water was found in the soil was low. And we also wanted to do the same exact analysis looking at the percent coverage of surrounding sagebrush and the sagebrush diameter. However, we did not find a relationship between those two variables and gall presence. We were also interested in understanding if these two factors influence the gall load found in infected sagebrush. So here on the left side, we have the percent water again, percent cover in the middle, and sagebrush diameter on the right-hand side. However, we did not find a relationship between these three factors and the gall load found in the infected sagebrush. We were also interested in understanding if sagebrush location influenced the gall presence. So on the y-axis, we have the proportion of absent galls and present galls on individual sagebrush, and in sloped areas and non-sloped areas. So that darker gray is where individual sagebrush that had no galls, and the lighter gray is individual sagebrush that did have galls. And we did find that in sloped areas, it was more likely to have gall infection than in non-sloped areas. So again, we were interested in understanding if the sagebrush location influenced the gall load on the infected sagebrush. However, we found no relationship between the sloped and non-sloped areas and the gall load.
So these results support our hypothesis and our initial observation. that sagebrush that grow in drier soils and on sloped areas are more likely to have the gall infection. Here we can see that the likelihood of gall infection dropped below 25% for soil that had 5% or more moisture content. We can also see that 80% of sagebrush that was sampled on sloped areas had a gall infection, while only 10% of sagebrush that was sampled on flatter grounds were infected. A possible explanation for this is that sagebrush that grow in arid soils and on slopier conditions are more stressed than sagebrush that grow in wetter soils and on flatter grounds. This suggests that the more stressed the sagebrush is, the more likely it is to have a gall infection. This interaction is important to understand for a number of reasons. One reason being that insect and parasite communities have been seen as an important indicator for the levels of health and biodiversity in an ecosystem. Our research can be used in further studies to better understand gall distribution in sagebrush communities, as well as to help scientists prioritize areas for protection and restoration based on their levels of health and biodiversity. Possible further research we'd like to conduct is if a sagebrush is infected, we want to know what factors contribute to the magnitude of that infection, so gall abundance. Another thing we'd like to do is sample and metals like Campido's metal to compare our results, sampling other subalpine metals to compare our results to what we found in Campido's metal. We like to look at north versus south facing slopes to see as well if those are contributing factors. And finally, we'd like to look at other insect communities to determine if there's a predator-prey relationship and if these other insect communities, and if that interaction plays into the whether or not galls are found on sagebrush and the magnitude of those galls found on sagebrush.
So with that, we'd like to thank the George Street Botanical Foundation for funding our research, the staff at Griffith Creek Research Station for housing us and letting us use their facilities, University of California, Santa Cruz for having us, and the George Street Conservation Scholars staff and other scholars for supporting us in all these items.
Does anyone have any questions?
You mentioned you looked at 2 gall sites. Did you see any patterns in those 2 or just
Yeah, so we did see a small pattern that we didn't mention in our findings. We saw that on our way towards the slope, towards the uphill, we saw that there were more of these woolly sponge midge galls that were found on like the flatter grounds, but as we got closer and closer to the slopes and the hills, we found that there were a major abundance in sponge galls instead.
Correct me if I'm wrong, but you guys only sampled Campido's metal, right?
Right.
And since that's like that, do you think if you had a wet meadow on a slope, you'd find anything different?
That is a good idea, but usually slopes are known for being more dry just because the wet tends to be at a flatter area. But that would be interesting to look at. I think it would have more or less.
I have a language question. Do you think that the north and south facing would kind of go along with that after the water content on the slope?
Yeah, so that was kind of like maybe what you were getting at, Alani. I think that would be an interesting thing to look at because one side of the north facing slope may be more wet than the south facing slope. So if you could look at more meadows and more areas, that's kind of one of the reasons why I mentioned at the end that we want to look at north and south facing slopes to see if there's difference in that and maybe tie that back to the water content in the soil.
To add to your question, other reasons is because like the sun both sets and rises in different areas, so that's also varying with north and south facing.
Hi.
So in the picture, I see like the Dolomite soil type at the top. Did you guys get that high? Did you ever overlap onto Dolomite soil?
No, that was a different slope. So we didn't actually, the slope that we measured on was kind of like on the other side of where they're at, but no, we didn't.
Yeah, one of the main reasons for that is because there was not an abundance of sagebrush growing in the Dolomite areas, and if there were sagebrush, they were growing under other critical environments.
I was wondering if you saw any insects on the gold, or like what kind of creature insects, if you know,
Yeah, there's definitely insects inside the gold. Cool. Yeah, so it was kind of, you guys did that some fun. Yeah, so it's two different types of midges. So yeah, so one was a doll midge and this one's doll midge. And so yeah, if you like get a knife and cut them open, if they haven't left yet, so usually if there's like holes in the gall, then they already left. But if you can cut them open, then like you'll see like eggs in there and like little insects in there. It's really cool. Yeah, so it's kind of another thing we were interested in, like kind of the insect species, and maybe also if we were to kind of go, like have an opportunity to go back and further look into these specific species and kind of see, and like learn more about their life cycles.
Was there any difference between your percentages of galls based on, it looked like it might be some open water where you were measuring, or was there any open water?
There weren't really like, oh, did you want to answer?
Yeah, well, Serena and I were measuring the sagebrush on the flat gradient, and so we actually didn't find any gold at all. Like where the soil was wet and where there was water, we found none.
Right. Well, I'm just wondering whether if you have standing water, whether that might impact, the distance of standing water, have some impact on
Right. There wasn't exactly standing water. It was more like mud puddles and like just super moist dirt. Yeah. But it wasn't really like a pond or anything like that.
But I will say to add on to that Serena and I went pretty far out. So first it was wet, and then it was somewhat wet. It was kind of damp, and then it was dry, and then it was wet. So it kept going, but the further out we went, like we didn't find any gold, like on the flat land that we were on.
Do they die from these infections, the plants?
That was my question too.
We haven't.
It's like if they have, I mean, we saw one plant that had like 44 galls, and it was looking pretty rough. Yeah. So I think they can die from the galls, but a lot of the times, like you'll find a sagebrush plant, and it's a really big looking plant. It looks fine, and then there's just like a couple of galls on them. So it's kind of, it would be interesting to like do a study or look into more of the research on like at what point does gall infection make them die. And so that was another thing when we were looking at this, we couldn't find any factors that predicted the gall load. And so it's kind of, there's some questions there. Once the plant is infected, kind of how long do they have, how many galls can they have before they die. So it's a lot of research for that.
Can I add to that? So something interesting that like our group really debated on showing in our presentation is that we noticed that when sagebrush were flowering, there are like less galls on it, but we weren't actually sure of the relationship between it being infected and whether flowering, like is it flowering because there's no midges, or is it flowering and then the insects kind of like infect it? So that was like a gray area.
Sort of like, is it the chicken or the egg that comes first? We just weren't really sure.
One more quick one.
Okay. Did you see like what size that the magnitude of infection varied? Like where bigger ones have less or more than smaller ones?
The ones Serena and I found didn't have galls, but I remember Carl and Julie saying that they found a really small sagebrush that had 44.
Yeah, it was smaller. Yeah, so it's kind of again another like interesting thing to look into, like is the relationship between like the gall load and the size of the plant, or the age of the plant and things like that. There's a lot of interesting things.
Thank you all for having us.
plants keep getting a little bit older with every talk here. We started with biennials, we went to perennials, and now we're going to jump right over to the oldest trees. And we'll Gustav, Kristen, Cameron, and Leslie to tell us about how alpine bristlecone and limber pine interact in the White Mountains. This one was the laser bit, and that one was the next one.
Hello, everyone, and thank you for joining us for our final Dorsey Conservation Scholars Symposium. My name is Cameron Oglesby. I'm Leslie Vargas from Muskogee Lake.
I'm Chris Clark.
And today we will be discussing our findings on the interaction and potential competition of bristlecone and limber pine tree recruits in the White Mountains of California. So, just so you know, this photo was taken by the marvelous Mushtak. In the White Mountains, the forest that exists at this elevation, which is about 10,000 plus feet above sea level, are blanketed by the dry, sappy bark of bristlecone and limber pine trees. These trees are the only species resilient enough to live in these mountains. Bristlecone is considered one of the oldest species in the world. Can you imagine? These gnarly pines live in 2,000, 3,000, 4,000 years. They are also home to a lot of interesting fauna in this area. It was the rare nature of these elderly beauties that drew us to study the surrounding environment and how it impacts these trees. So the two primary tree species, again, bristlecone and limber pines, can be distinguished by the following characteristics. Bristlecones prefer nutrient-poor dolomitic soils, surviving in conditions too harsh for most other tree species. They're specialists, persisting in a narrow elevation range for long periods of time. Limber pines, in comparison, are generalists, surviving in a diversity of soil types and nutrient levels. Their elevation range is much broader, between 5,000 to 12,000 feet above sea level. To provide a larger context, numerous studies have pointed to a domination of bristlecone forests by limber pine recruits. As climate change shifts both of these species higher in elevation, the migration and recruitment of limber pines outcompeting bristlecones, presenting the possibility of limber pines overtaking alpine and subalpine ecosystems. Such forest composition shifts have major implications for biodiversity that relies on bristlecone forests for security, food, and shelter. For example, these forests are a major support system for several songbird species in the area. These forests are an important and long-lasting source of carbon sequestration and storage. And the biggest point, there is no alpine forest without the bristlecone. They are the heart and soul of the So our larger question, our big question, is whether or not an increased presence of limber pine recruits impacts bristlecone recruits. Looking to see if the direct competition for resources and space impacts bristlecone settlement.
Okay, so our first question is posed up as A, so whether or not the presence of limber pine affects bristlecone, and so we're kind of expecting to see that it will. So more limber pine, less bristlecone recruits. Our second question was then, okay, does dispersal vary between species across soil types? So we're kind of hoping to see that there will be more limber pines in most of the soil types if they are in fact kind of taking over. And then our third question was, does the relative abundance of size classes differ across the sites? So are the sizes of the trees different? Are we having more bigger ones in certain soils? And so in order to answer these questions, we picked 3 sites with 3 different types of soils. Our first one was dolomite on the top left, and then we did granite, and finally sandstone. And at each site, we did 10 plots, each 1/10 of an acre big, and they sort of look like this. So we would start with a center flag and kind of pick a spot, center flag, and then we would do an 11.3 radius. from that center flag and set up a quadrant. We would then have someone like standing in the middle of the circle and taking notes while the rest of us went to number 1, identify trees in order to set them in their categories. We would then do diameter of breast height, or really kind of the circumference, and then we would calculate diameter later. We would do cone bearing, and this was kind of to separate whether or not they were mature or juvenile. So with cones, they were mature, without juvenile. And if they were juvenile, we would then do distance to mature trees, kind of to see whether that dispersal was happening. And it was really simple, distance. And so these are just a few pictures of kind of what we did. So this is Chris doing DBH. And so he's kind of hugging the tree, and then we're measuring that, calculating the tree DBH later. And then in the second picture, we have Michelle holding the transect tape while we were setting up our plots. So she's in the center, and Cameron's all over there. She's running and getting, probably was, and she's getting the end of the plot, and we would do this about 3 more times to get that full quadrant.
All right. So the first question we asked was, does the presence of limber pine affect bristlecone pine or fruits? These are actually the data points that we collected. On the x-axis, you can see percent limber pine from 0 to 1, and on the y-axis is bristlecone or fruits per 400 meters squared. That's the size of the area of the plots. So this result was hypothesized to be a negative slope, which when you do a line of best fit, it looks like, wow, we did that. But then, if you separate it into soil types, you realize that that line doesn't really exist. So this bar chart kind of shows clearly why, because the percent limber pine was really affected by the type of soil you were in. So much so that you couldn't really accurately predict or say with any certainty how many bristlecone recruits, if the number of bristlecone recruits was being affected by the percent limber pine. In dolomitic soils, you have much less limber pine than you do in granite and sandstone, which makes sense because if we give it its natural history, limber pine prefers granite and sandstone. Okay, so now that we know that soil type rather than the presence of limber pine is actually the driver of forest composition, the next question we asked was whether or not dispersal varied between species across the soil types, because our background research suggested that limber pine was a better disperser. So the way that we did this was, on the x-axis we have the different types of soil, and then on the y-axis we have the distance from nearest adult. So in the field we would find our juveniles or non-cone bearing trees, and find the distance to the nearest adult tree of the same species. When you do statistical tests on all of these, you find that the means are not statistically significant across the different species. So dispersal doesn't vary between the species across the soil types, which is contrary to what the research suggested. This is a slightly different graph. It's kind of just ordering things differently. We look at each species, and then across the different soil types, we see whether or not the distance from the nearest adult changes. This is more like asking, does for bristlecone, does the dispersion from adult tree change between granite, sandstone, and dolomitic soil? When we did an ANOVA for bristlecone, we did find that the means were different. So this is kind of an area of potential further research. Maybe dispersal is affected by the type of soil that these trees grow on. Okay, so soil, important. So one of the questions we looked at was whether or not the relative abundances of the size classes was different across the soil types and species. The histogram kind of shows you the frequencies of different diameters at breast height in meters of the different soil types and the different species. You can see some interesting trends here. In the dolomitic soil, for bristlecone, you're more likely to see larger trees than you are for that species than granite and sandstone, and the opposite happens for limber. You're more likely to see larger limbers in granite and sandstone and not in dolomite. So the smallest trees are found in the soil that you don't prefer.
Okay, so in case you've been asleep for the last 3 minutes, soil is the underlying driver. Cool. So how does that relate to the bigger picture? What's next? Okay, so first to kind of explain partially why soil might have acted the way it did in our experiment. Limber pines, much more so than bristlecone pines, need water and nutrients, especially in the spring and summer seasons when precipitation is less. Dolomite is notoriously harsh, low nutrient, which is why we saw some juvenile limber pines with no adult ones, so perhaps they can establish in earlier months when there's water, but they can't grow. This, on the flip side, points to dolomite being kind of a refugia for bristlecone pines because we didn't see them too much elsewhere. So we kind of need to keep that in mind when planning management actions for the future, that dolomite is this specialty niche where bristlecone pines thrive, and we need to sustain that for the future. Okay. In addition to that, we had some cool fauna that we saw. The bottom picture is, okay, it's a Clark's nutcracker, and Ms. Clark also took that picture. And then the top picture is a pinyon jay, which admittedly, you can tell by the name, they're abundant in pinyon pine forests, but in the Inyo National White Mountain region, they really depend on bristlecone pines. And on the IUCN Red List, they're listed in the threatened category, subset vulnerable. So, and there's only known for the genus, which I thought was just a cool fact. So they really need to really manage these bristlecone pines. We want to see pinyon jays still existing in the Inyo National Forest. And then, just wrapping it all up, how will climate change things? This is kind of an infographic of how snowpack in the Sierra Nevada region will change under lower warming and higher warming climate range estimates. I've kind of put the White Mountains area in the box and an arrow. But as you can see, by using the color scale on the bottom, there's going to be a less range of snowpack and also less water in the snow, or less snow falling from the sky. And there's going to be less water. There's going to be less water on the ground on April 1st. Which is the main takeaway, which means for both these trees, that's less resources, less nutrients available for them, and especially for the pinyon pine, or for the limber pine bristlecone pine relationship, more water will fall as rain in later months, perhaps exacerbating the competition and the exclusion of bristlecones from the native habitat and allowing limber pines to kind of overtake that niche. And bristlecones are important. We've talked about, the carbon sequestration because they live for thousands of years. We've talked, and they're very culturally important. That's the only reason anyone will ever go to the Inyo National Forest, ever. There's nothing else there, right? They're important in a lot of ways. And yeah, so we kind of need to monitor this as climate is changing to preserve these very important trees. And with that, we'd like to thank everyone, the Dorsey Charitable Foundation, this program, all our scholars for the support, riding with us. If you have questions, we'd be glad to take them. Yes.
Cool that you found those bristlecone on sandstone, but that they were all little ones. So what do you think is going on here? Do you think it's that the bristlecones are starting to invade into the sandstone, or do you think it's that every time a bristlecone gets started in the sandstone, it dies? Birds.
Talk about the birds?
Something, actually, yeah, people don't really know how bristlecones disperse. Like there's some theories about whether or not birds spread seeds over there. So yeah, it was interesting for both lumber pines and bristlecone pines. If you want to look for young trees, you actually go to the soil type that they don't like, which might just be kind of like a bias because these bristlecone pines may not survive. But at the same time, both bristlecones and lumber pines have like pretty low mortality after a few years. So like if they can survive to a certain point, they're guaranteed to not guaranteed, but guaranteed to live a long time. So kind of a mystery.
There was also a paper that I read that said that bristlecone seedling survivorship is higher the further away they are from bristlecone adult, which may answer maybe.
In terms of they're actually recruiting on sandstone, we learned when we went to meet the park ranger, John, I believe his name was, that they can survive on other forms of soil, but they do best on nutrient-poor dolomitic soil because they can easily get outcompeted. So seeing higher densities of smaller bristlecones on the ones they don't prefer is just because they can. They might not necessarily survive there.
Do you guys think if you added the moisture content in the soil, that would give you guys steady warming after future? Or do you think it would be more about the plant material and stacking in the water?
Oh, yeah. So I think moisture content would have been an interesting thing to look at. Just based off of your question, I think in order to have it for it to have broader impact, we have to look at it over years to kind of see how snowpack and precipitation that falls as rain in like the April to September-ish months is changing, and then how that soil moisture is changing, and then how the creamer is changing. But I think it'd definitely be something to look at.
We also thought about possibly doing maybe like pH and kind of doing the nutrients of the soil as well, but with our time constraint, it's kind of hard. But that would be something cool to look into.
Was either species easier to study than the other in terms of like actually going in there and really questioning the pH and stuff?
So personally, I speak your piece. All right, I've measured a lot of trees. I'm the forest girl, right? Limber pines have these little gnats that like just fall and stay on your body for a few hours, and it's awful. So limber pines were not the best. Bristlecone, they're all kind of old and they break off, so it wasn't that hard. Not that we tried to break them off, but you know.
I will say that there were a lot of these, I don't know if they were bullet ants or what, they were all over the bristlecone. They bit me. So I preferred the limber pines. They weren't as Costa Rica.
But painful. I guess we all had our personal preference.
Do we have time for that now?
I think one more quick question.
You can get somebody else.
Desiree.
What's the main difference between limber pine and
Between the two? Oh wait, can we go back? Oh, okay, these are the key differences in terms of identification. As Moonpath said, limber pines tend to have a lighter bark, more light-skinned trees. Whereas bristlecones had reddish cones on the end. They tended to be more twisted. But these are the key life history differences. Identification is more bark color, cone color, and shape. Those sorts of things. Hope that answers.
Okay, thank you.
All right. Scheduled to take a short break now, so folks can stretch, use the bathroom, grab some refreshments, and think really quickly. We're going to take a break for just 15 minutes here and come back. Oh, we're done at 2:10. And make sure you come on back, because we're going to talk about lupins, and then bristlecones again.
and I have learned so much from each and every one of you. And these presentations have been amazing, and I'm really looking forward to the second half as we continue our journey through the White Mountains. So without further ado, I'd like to invite up Cameron, Rebecca, Paloma, and Omar to talk about effects of lupin sagebrush as hosts on paintbrush in the White Mountains.
Hello, everybody, and welcome to our presentation on the effects of host and competition on desert paintbrush in the White Mountains. And before we start our presentation, we would just like to acknowledge that the White Mountains of California is an area of cultural significance to the Bishop and Big Pine Paiute tribes.
I'm Rebecca, I'm a building.
I don't like colors.
I'm Cameron Howell.
And I'm Paloma Marco. So, for our introduction, we just want to make everyone aware that desert paintbrush, or kasupi in the Big Pine Paiute Valley dialect, is a parasitic flower. And we really wanted to investigate the question of what drives the success of parasites from the parasite-centric view of the paintbrush flower. Is it the host, or is it competition that determines success? Is a parasite considered more successful based on who it is parasitizing from? Or is it considered more successful when there are many parasites on the same host? Also, we want to look at this subalpine environment because it has unique conditions, being that it is in the rain shadow of the Sierra Nevada mountains and receives less mean annual rainfall and has higher wind and snow conditions during certain times of the year.
Okay, so to give you some more background information on the plants that we focus our study on. So we focus our study on the parasitic plant known as the desert paintbrush, which is native to the California White Mountains. So it's a heavy parasitic plant, which means it's only partially parasitic. So it takes some nutrients from its host plant, but it doesn't necessarily depend on it for survival. It actually only depends on its host plant during its flowering period. The way that it parasitizes its host plant is through the use of hysteria, which are like slim protrusions that it has on the root system, which it uses to pretty much dig into the other root system of its host plant and pretty much just suck the nutrients out of the plant. It's illustrated in this picture. Yeah, so we want to investigate the success of desert paintbrush in the presence of two different hosts. That being the sagebrush, which is a common brush that is found all throughout the White Mountains, kind of like a dominant plant everywhere, and the lupin flower, which is a nitrogen fixator. So we thought that the lupin would be more likely to be the host because it's a nitrogen fixator, so it would probably provide more nutrients for the paintbrush if it was to parasite.
So this leads us back to our question of understanding the background of the host and the parasite. And it makes us ask, does the host community or competitor community improve the reproductive success of paintbrush? In a previous study that was conducted in Colorado and Texas, it was found that paintbrush flowers that were parasitizing on lupin showed double the seed count and less signs of herbivory. And this was significant because these areas where they found the most productive paintbrush were at lower elevation in sunny, moist areas. And we wanted to see if this same relationship held true in the subalpine environment of the White Mountains. And we assumed that the lupin and paintbrush would have the same relationship, and that lupin would be the optimal host
improve reproductive success of paintbrush, and that competitors of other paintbrush parasitizing on the same host will reduce the reproductive success of the paintbrush parasite.
Okay, so we collected 20 paintbrush samples at 3 different locations in the White Mountains. We chose these locations because they had the same substrate for soil and they were found on similar elevations. So for each sample, we collected the 3 tallest stalks of each paintbrush and we collected the total number of flowering stalks. And we did this in order to have a representation of the reproductive output of the paintbrush. We then used, as the diagram shows, we then used a circle plot, a 2-meter radius circle plot, to calculate the percent coverage of sagebrush and the percent cover of lupin surrounding an individual paintbrush, and we also counted the number of individual paintbrushes in the circle to determine the density of paintbrush surrounding an individual paintbrush. We then used a linear regression to understand the relationship that paintbrush density and the percent cover of lupin and sagebrush had on the reproductive output of individual sagebrush. And this picture is just an example of one of our sampling sites. As you can see, you have the lupins, the paintbrush, and you have sagebrush in the same community. And this indicates our 2-meter circle plot that we use. So we did, of all the transects, we recorded the percent cover of sagebrush and the percent cover of lupins. And within the circle, we did each individual paintbrush. And these pictures are just to illustrate the variation in reproductive success in paintbrushes, as seeing how stalk heights vary greatly, as well as the number of stalks for individual paintbrush can vary greatly depending on different paintbrushes.
So when we looked at our 2 success-contributing factors, which are stalk height on the left and number of flowering stalks on the right, we quickly discovered that there is no relationship to lupin percent cover, and that reproductive success of paintbrush is not significantly improved by the abundance of lupin, which kind of feels for a loop because this was contradicting both our hypothesis as well as the findings of several other studies done in other environments. And so this caused us to pivot and reconsider within our environment what could be most affecting the reproductive success of paintbrush, whether that was other potential hosts or competition, competition. So then we looked at sagebrush, and we discovered that the mean stalk height of individual paintbrush increases by 2.14 centimeters for every 1% increase in sagebrush coverage. And we found that individual paintbrush actually do better in the presence of increased sagebrush hosts. And this is great for the individual paintbrush. However, when we looked at the community of paintbrush, we saw an increase in paintbrush density as sagebrush percent cover increased. And so in conclusion, we were able to see that paintbrush density is lower, but individuals are taller when there's a higher percentage of, or a higher percentage of sagebrush. However, if sagebrush percent cover is lower, our individual paintbrush do worse, but the communities do better. So this caused us to consider that perhaps interspecies competition may be a more significant factor to success. And what we found was that paintbrush stalk height actually decreases for the individual as paintbrush density per meter squared increases. Which was great because our hypothesis that interspecies competition is a driving factor in individual paintbrush success is occurring in this design.
So kind of wrapping things up in full circle. Yeah, so going back to the study I was mentioning previously, so in the study that was conducted in Colorado, they found that the paintbrush do benefit from the lupin over other hosts. Our study, however, showed no correlation between paintbrush reproductive success and lupin cover. Some possible reasons that we talked about previous was that the nitrogen levels in the soil may have been insignificant. So the paintbrush may not have had a preference for the loop end because there was just not enough nitrogen to really make a difference. The reason for this could have been that the decomposition rates in the soil were just too slow, and there's just not enough nitrogen, and there's just not enough moisture in those types of environments. What we did find instead is that subalpine environments, paintbrush resistance success is improved when there's a high percent cover of sagebrush and a lower density of surrounding paintbrush. This may suggest that sagebrush is an optimal host, and that interspecies competition is what's driving the success of parasites in this environment. And while the host preference of the paintbrush is not known yet, we recommend that future studies focus research in a controlled environment to really see if there's actual preference in different hosts that the paintbrush chooses to transfer. And to pretty much wrap things up, what we found from our study is that in these extreme environments in the subalpine, interspecies competition is a true driver of the determined success of parasitic plants, and it's not the host that it chooses to test.
We'd like to give a big thank you to the Australia Foundation as well as the Genesis President, staff, and co-hosts and the support and guidance of all this whole process. And thank you all for being here with us today.
If you were to conduct further research on this experiment in a controlled environment, what would that environment look like?
We'd probably do it in a lab and just kind of this quick idea we had when we were getting ready for this presentation. Would that it would be interesting if put the two hosts in the soil and then put the seed of the paintbrush and then see which one it goes to in closer proximity and kind of use that to determine the pressure.
And to elaborate, I think that it might also be better to use a more mature paintbrush too to see if the roots parasitize later in life besides once the seeds first germinate.
You mentioned it's only parasitic when it's flowering. Is there any evidence of competition with the host plant when it's not flowering? But when it's growing on its own? Well, it was in other studies on paintbrush. It was found that they could photosynthesize on their own. But I think that if they were on their own, we would see differences in the stalk height, the number of flowering spots, and kind of bring it back to the competition, if it's just one or if it's multiple ones. Do they do better with or without? Just to add on to that, the relationship between paintbrush and the host is really complex. And so while we can survive on its own, they do a lot better than they are parasitic. And so that is specifically helpful in maturation. But in a lot of other studies we found the relationship is really complicated and also has linked to reduced delivery, as you mentioned, and the female fitness specifically as well. So yeah, I agree. And also referring back to the literature, the mechanism that it uses of bacteria, sometimes they found that it'll kind of root itself onto the sandy pebbles and substrate, and so that also kind of further complicates the actual behavior of the
Did you determine reproductive success by how many plants were in a particular area?
We were using I don't know if explain that, but we used the number of plants as an indicator of competition, interspecies competition among paintbrush. So that's why we did the number of paintbrush within the area to see if they were competing for the same resources.
that was reproductive success?
That was more to look at the community and the competition, but for the reproductive success, we looked at the average height of the seed stalks and then the number of flowering stalks on that one.
Sorry, one quick question. I like that you mentioned the previous studies that happened in other states. I was curious about what were the differences in methodologies between your study and their own.
Time was definitely a big factor because the study in Colorado and Texas was done over the course of 2 years, and in ours it was over the course of 5 days. Time was a big factor in that study. But I think that they looked at different factors for reproductive success, like they looked at the seed count, and that's something that we didn't include in our study because of the time of year that we were there. So maybe going at a different time of year and seeing the actual seed count maybe after they're perennials, so the next year when they're coming up, and in that time frame when they're regrown, see if that is specific.
They also determined whether or not the paintbrush was parasitizing on lupins by collecting the leaves and then conducting nitrogen samples to measure that nitrogen uptake, which we tried, but our results weren't really conclusive.
I'd like to welcome Maya, Claudio, Eleni, and Desiree to talk about reproductive success of lupins and different aspects of the White Mountains.
Good job, Joe. I've had to try to yell at people, man.
All right, welcome everyone. Thank you so much for being here. My name is Eleni Oka.
I'm Desiree Ponte. I'm Maya Gaines.
Claudio Maya.
And our project is on the reproductive success of the silvery lupin in the White Mountains of California. So the White Mountains of California are 7 mountain ranges east of the Sierra Nevada. The White Mountains hold a subalpine environment where temperatures are lower and substrate is much drier. So with climate change, global temperatures are increasing drastically, affecting higher elevation habitats by increasing snowmelt, which results in more moisture distribution to species in the wetlands, including to the silver lupins. The silver lupins are native plant species to California. This plant is considered a perennial plant due to its year-round existence. This plant is also a nitrogen fixer, which is essential to other plants to thrive in that area. In fact, lupins coexist with sagebrush and Indian paintbrush flowers in the dry sandstone substrate. Additionally, silver lupins are alkaloid-rich plants, meaning that it can be fatal once it's been ingested. So, for example, cows have been reported to have died from eating lupins. And lastly, these lupin plants in the White Mountains are home to the endangered blue mission butterflies. So our study focuses on the lupin interactions with different aspects in the White Mountains. Because the lupin plays such an important role in subalpine ecosystems, we designed a study to investigate what factors affect the reproductive success of silvery lupin in the White Mountains, in hopes that the answer to our question can be applied not only to the general subalpine environment, but to also inform conservation action for the species reliant on the plant, such as the mission blue butterfly that Desiree mentioned. We measured reproductive success by using percent developed seeds, developed meaning
pollination and fertilization occurred. And we also had an additional measure of reproductive effort using number of inflorescence, which is just flowering stalks on the plant. We hypothesized that as elevation increased, the number of inflorescence and percent developed seeds would decrease because at higher elevations there are lower resource availability. And we also hypothesized that north-facing slopes would have a lower number of inflorescence and a lower percent of developed seeds because in the northern hemisphere, north aspects not only receive, or because they receive less direct sunlight, they have more moisture, and in environments as harsh and dry as the subalpine ecosystem, moisture is one of the main influencers of plant community distribution. Our study used 8 sites, so we had 4 on north-facing slopes, 4 on south-facing slopes at various elevations between 10,000 and 11,000 feet. Each site had 2 15-meter transects that were placed 15 meters apart, and we randomly selected either the left or right side to sample. So whatever selected side, we then sampled every individual within 1 meter of the transect on that side, and we counted the number of inflorescences per plant, and then we also collected at a maximum 5 seed pods per plant, and then calculated percent developed seeds as an average. And we had a total of 210 individuals, with 108 individuals being on north-facing slopes and 102 individuals being on south-facing slopes.
This diagram is a representation of what was stated before about the south and north-facing slopes. As Maya mentioned before, you can see south-facing slopes actually get more direct sunlight. That's the reason they're actually more dry and they have less moisture content in comparison with north-facing slopes, which they tend to have, they tend to be more wet, and then they tend to have more moisture, and also they tend to be more nutrient rich. So this key factor is actually very important for plants to determine where they're going to be located. So our results. Over here in the y-axis, we actually have the bio we talked about earlier, which is the percent for the developed seeds. And in the red axis, we have the elevation belt, which is what we work on, between 10,600 to 11,000 feet of elevation. And the different, the 2 lines actually, one of the red line represents the south slope, blue one represents the north slope. And you could tell there is like a slight negative trend in which as elevation is increasing, right? We actually see a decrease in the number of, in the percentage of developed seeds. And it's basically, the plants are actually, the higher the plants are actually going, the less it's growing. And the other one, the variables are the same. The only variable that's different would be the inflorescence for square meter, which is what we determine to be the number of inflorescences. And the trend that we see, we actually see a more strong pattern, but only seen with the north slope. It's basically as the elevation is increasing, then we see an increasing of the number of inflorescences.
So our results showed that with the increasing of elevation, there was also an increasing of the number of inflorescences per meter square on the north aspects. And we also found that there was a slight trend that with increasing elevation, there was a decrease in the percent of developed seeds also in the north aspects. And so our research actually had a lot of contradictions with other research done in high elevation on plants. A lot of other studies found that plant growth rates tended to decrease as the elevation increased, which is the complete opposite of what we found in the northern slopes. And then after doing more research on the lupin plant, we actually found that lupin plants are very drought resistant, and they normally grow in dry, hot aspects such as the southern aspect. So it was really interesting to us that the northern aspect actually had a lot more inflorescences.
But we believe that because the northern aspect has more moisture, it has more resources and more water availability. And because of this, the plants in the northern aspect actually reproduce more seeds because also in other studies they found that there are less established seedlings in higher elevations, and maybe because since the northern aspect has more resources, and they also know that not many of their seedlings will survive, they reproduce a lot more seeds, which gives them a better chance of survival. We also thought that, sorry, going back to the rain shift, since climate change could affect the elevational shifts in these plants, that will also affect the species that rely on them, which include the blue-mission butterfly. So if these plants decide to rain shift, the blue-mission butterfly will be taken with them. And that could actually cause a lot of disruptions in other environments that are higher than the leaf and altitude range, because all of these new species interactions could possibly alter the communities that are already there.
And with that, we'd like to thank the White Mountain Research Center for allowing us to conduct our research and also giving us a place to stay. We'd like to thank all of the Dorsey Conservation Scholar Program staff for helping us with our statistics and also providing us some much needed emotional support at times, because the White Mountains was really rough for all of us in the team. And then lastly, we'd like to thank the Dorsey Charitable Foundation as well as UCSC Dorsey Conservation Program for funding, and a special shout out to Eric Zavaleta and Justin Cummins for honestly just allowing this whole thing to happen.
Any questions? Sorry, I'm not like a plant person at all. I understand why one side would have more water, but why would one side have more nutrients? Why would one's low?
It has to do with the so in a dry environment you're gonna see less amount of plants because plants, more plants tend to like plants like water. Yeah. So if you don't have that much water in the other fields, they won't like to be in that and in the other fields. And then most actually there are some plants that they tend to make the soil more nutrient. Mm-hmm. Like they tend to like the lupin. One thing that it does is a nitrogen fixer, which basically increases the level of nitrogen into the soil. That's why we have like HPAC and stuff like that. And other plants do other things like increasing the level of potassium, level of calcium and phosphorus, which is very important for the plant. So the more plants you have, if you have water into one of them more than the other one, more plants are going to prefer that.
Thank you.
So, I don't understand that much about this, but just curious, why not look at all of the seeds that are present as opposed to just developed seeds? Is there no chance that the ones that aren't developed now could become pollinated or
I'm going to answer that question to the best of my ability. And maybe, Nikki, if I'm incorrect, you can steer me in the right direction.
When we started this, so when we started this process, we were under the impression, if you look at the photo at the top, it's actually really hard to see the undeveloped seeds because they're they're so tiny, and then the developed seeds are obviously the larger seeds. But it's my, I guess, interpretation that those tiny seeds, they're never going to get there, you know? It's too late. And maybe you're saying that's correct.
To add to that, we actually count that too. We have in our data we collected a lot of data, and we did actually count the undeveloped seeds as well.
Do you think the weather this last season, which was rainy, might have had some impact on you or having different results than what was present in the literature?
I think it definitely could have.
but we also didn't measure that, so I can't say if it would have or not.
So the butterfly, its larva lives in the lupin plant, from my understanding of it. So it'll lay its egg and its larva will come out, and then it actually uses that plant also as a food source. So they're endangered. So they kind of need their food sources.
Yeah, I'm curious how long-lived individuals the lupin species are, and the reason why is that long-lived tree species in other work in the mountains over the last century, in some cases, have shifted from being more successful at high elevations on South Facing aspects to being more successful at high elevations on North Facing aspects, because it's gotten enough warmer that the North Facing aspects have stopped being moving. So I guess I'm just trying to figure out how much improvements are like trees, are they 50-year plants or are they 10-year plants, and I don't know if you know.
We actually didn't collect the data related to that. One of the reasons, we actually measure the size of the plant and the length that you have, but that's not a reliable measurement for age. And then we read a paper on that too. So we really didn't have the time, the tools in order to measure, and we didn't really know how to take it.
To round out our symposium, I'd like to welcome Nautica Jones, who will be talking about bristlecone on north and south slopes and how that may be indicative for our future.
Welcome to my talk. Thank you for coming. I'm aware that right now may be a hard time for a lot of you that are here today, and I appreciate you sort of taking the time out to come here in this special time for you all. But today, we're going to talk about the future. Wrong clicker. We're going to talk about the future because something's coming, and in fact, it's already here. And it's not winter, it's warming. So the warming that I'm talking about is actually part of a much larger a threat called climate change, and it's affecting not just one system or one species, but it's affecting all systems and all species, 1 million of which may face extinction due to this problem. And you might be wondering, how is a little bit of warm weather going to cause one species, let alone 1 million, to go extinct? Well, the answer is that it does it indirectly. You know, climate change isn't going around directly competing with anything or spreading, or it's not a disease that's going to kill off some population, but it is causing indirect changes like phenology. It's lengthening and shortening certain seasons, making temperatures more intense in different seasons. And in some cases, it's melting a lot of ice that's causing sea level rise and habitat loss for animals that will depend on that area. And among those things are contributing to rain shifts. And so these animals are leaving these once suitable areas to find new areas that are going to be more conducive for them. But you could probably figure that there are already animals, there are already species there. So you're getting an increase in competition as well. And with any competition, there's winners and there's losers. And for some species, that loss is going to mean local extinction. And one species we don't want that to happen to are the bristlecone pines. Now, you may be thinking to yourself, you just said around 1 million species may face extinction due to climate change. We have lions, we have tigers, we have bears. Why are we talking about trees? Why are we talking about this tree? What are we really going to lose if this tree goes extinct? And you're going to lose about 10,000 years of insight. Within the tissues of these trees, the oldest living organisms on the planet right now is over 10,000 years of natural history data that has already gone on to inform our decisions and what we know about climate change. In fact, we have a dated timeline of warming that has been derived from these tree cores. And I expect that even more of our decisions in relation to management, policy, and human adaptation to climate change can be informed by information from these trees. So, now that we care, what are we going to do? We can't see into the future, but we may have a way to mimic the future. And that's through north and south facing slopes. So, what we know about north and south facing slopes, this picture looks very familiar. There was no collaboration. Except with Google. Anyway. So we have these south-facing slopes, which are receiving a lot more sunlight than these north-facing slopes. And so the south slopes are warmer and drier in comparison, meaning that they're not as preferred by the plants as, say, the north-facing slope. And so what we are hoping, well, what we can see from these south-facing slopes is that they're predictive of what a warmer future will look like. And so we phrased this question, how does bristlecone abundance vary on these north and versus south slopes? So this is how I did that. What you're looking at is a picture, a satellite picture from Google Earth. It's zoomed in, and you see a line, a red line going across called RL2. That is a transect line that is drawn over a ridge line. And so there are 4 transect lines, so there are 4 ridge lines in the study. And I sampled in circles on both sides of the slope. So there are 50 meter radius circles on either side of each slope, and then I counted the trees. And the reason why these trees are red, because they're not red in real life, is that there's a false color infrared layer on top of this to make the individual trees easier to count. And so what we found is that there are a lot more trees on the north slope versus the south slope. And I can tell you when I was looking at this, I became very discouraged. I became very afraid of what the future of these trees would be if the south slopes are going to be truly indicative of the future. And that prompted a follow-up research question. That prompted a question asking about refugia. Are there places on these south slopes that can house a more robust plant population, one that's more similar to the north slopes? And so, actually, before we go there, the way I asked this question was in relation to aspects. So I don't think this pointer is very bright. So, so in relation to aspects, so this is the south slope. So whether you're on the southwest or the southeast, are there more trees as you go more east or more west? Or are there more trees in relation to elevation? So whether you're on the more higher in elevation or lower in elevation on those slopes. Okay. So here are 2 graphs. And this graph closest to me is about aspect versus number of trees. So on the x-axis, you'll see aspects labeled southwest, south, and southeast. On the y, you'll see number of trees. And then at the top, there's a dotted line. That dotted line is the mean number of trees on the north slope. And the gray rectangle surrounding that is the 95% confidence interval. And then on this graph, Everything's the same except for the X, which is elevation. So what I saw in looking at aspect is that aspect doesn't really describe any refugia for these trees. We see a lot of trees in the southwest, few trees in the southwest, a lot of trees in the southeast, few trees in the southeast. It doesn't seem like aspect is very important to these trees. If they're already on the south slope, you're south and that's it. I'm from the south. But when we looked at elevation on these south slopes, we see that it's almost as if the trees do better on the periphery. We see a lot of trees either at the lowest extreme, and then we see a lot of trees at the highest extreme. And so this pattern is very suggestive, and we think that if we went on to sample further, that we would see a stronger relationship between that. And hopefully so, because we would like there to be some refugia through these trees. And the reason that we included data from these north slopes is that that is our reference for what we're going to call refugia. So the north slopes had a lot of trees, and so if the number of trees in the south mimics the number of trees in the north, we are calling that refugia. So what does all of that mean? It means, for one, that the future of these north slopes may look a lot like the current state of these south slopes. But we do have some hope in that maybe there is refugia in either these low and these high elevations on the south slopes. So there is some hope here. And so here are some a little bit broader implications. And if we can identify these places we expect to be refugia, we can protect them in advance. We can make sure that we give them the best fighting chance at surviving and adapting to climate change as possible if we have these areas set aside. And then knowing that there is going to be some effects from climate change on both of these slopes, we can expect to see more stress on the species that depend on these forests, as the forests themselves are going to be stretched. That is food, that is home, that is safety for a lot of species, and so we expect them to be affected as well. That is the end of my presentation. Thank you for everyone who helped me, for all of the support. I got a lot of support. Are there any questions? Maria?
So for how you use Google, can you tell us more about how you use Google Maps to conduct your experiment in terms of like how you got like the sound aspect and like elevation and stuff like that?
Yeah, sure. I'll bring you back to the picture. So I used some satellite images from Google Earth. But the website that I actually did all of the research on is CalTopo. So if you go on CalTopo, there's an option to use the satellite pictures from Google Earth. And what you do is do you want me to tell you how you search and find the place and then you
No, just like what you're able to do or not.
Oh yeah, okay, so I was able to draw onto the pictures, and it was able to create polygons for me and places where I can sample. There were a lot of filters and layers involved, like for example on this picture, all of these lines that you see, lines like this, not like this. These are elevation lines, so it can give you a lot of data about where the sun is. You can go back and forward in time. You can see where you can get a lot of information. Yeah. Other questions? Kyle?
One of the things I just know bothered bristlecone pine is their inability to compete with other species. I was wondering if you noticed an increase or a decrease in the number of species that would have to compete with on either side or no?
So where I sampled is on dolomite, and so there aren't a lot of other species on the dolomite. The higher you go, the less other species you see.
Okay, so that,
So there was little to no competition with other species.
so that high elevation dolomite refuge may exist.
Yes.
Awesome.
Cameron?
Was there, okay, we might be the same answer that you gave now. Was there like any way to control for different species or since it was on the dolomite soil, all the species that you saw from the map were pretty much?
Yeah, the dolomite I would call a control because there just weren't any other species at where I was. If you go lower on dolomite where you're transitioning into other soils, then you will see other species, but where you are on like exclusively dolomite, there aren't really any other species. Yes?
Were there differences in age groups of the trees, like was there a lack of a certain size or age group on the south side that you found higher representatives on the north side?
Yes and no. So you actually can't really tell from this picture, but it was generally seen that there were bigger trees on the north slope. There were more big trees on north slopes than there were big trees on south slopes. A lot of the trees on the southern slopes were smaller, but I've also learned that you can't really date the trees very well based on size. So I can't say if there are older trees on one slope rather than the other, but there are more big trees on one. Hi.
Have any hypotheses yet for why the high elevation and low elevation not very relationship?
I have a guess. In terms of the higher elevation on south slopes, I would think that's because the higher in elevation you go, the cooler it is compared to if you were lower in elevation. But there isn't like a huge gradient in elevation on one slope. Like, see how small this slope is? Like, it's not tens of thousands of feet in elevation difference. So I don't know how relevant that is on one slope, but I would think in a whole range that would be more important. Other questions? Okay, thank you.
Thank you to all our scholars that gave an amazing presentation today, and I'd like to invite Justin up to have some closing remarks.
So this year we received about 250 applications to this program, and throughout the month of March, our staff went through these applications and evaluated the applicants and chose this amazing group of 20 people to join us this summer. And just to give you all a sense, about 8 weeks ago, all of our scholars arrived here on UCSC campus. And we spent these 8 weeks really learning about professionalism, about research and conservation, and about the different issues that we're facing related to conservation. And so we traveled to a variety of different sites throughout the state. We went down to Big Sur, Point Reyes. We spent time in San Francisco. We went to Yosemite, the Eastern Sierras. As many of you have seen, we spent some time in the White Mountains doing research, and then the scholars all came back here where they were able to analyze all their data and kind of get ready for this final moment, which culminates the first year in the Dorsey Conservation Scholars Program. And so I want to start by thanking all the scholars for applying to this program and for choosing to join us in this experience. Really hope that you all take a lot away from this, and as this year goes forward, we're going to be working with the scholars to place them in internships next summer where they can get real on-the-job experience in a variety of different fields. So some of them may choose to do research, some of them may want to go into education, but it's just another way that we can help prepare the future conservation leaders in our field. And then after that internship, we'll have a week-long winter workshop to kind of wrap up the formal portion of our program. But before we go there, I just want to also express on behalf of the program, Erica Judy Straub, who just joined us as our internship coordinator, and myself, we spend a lot of time all year really trying to pull the pieces together and work with our partners and other people that we know so we can make this experience happen. But it really wouldn't be able to happen, especially this summer, without thanking a number of people who I just want to mention right now. And I'd actually like them to stand if they're here, but there are some people who I'm going to thank who aren't here. And I'd like to start with our instructors. So I'd like to thank Dan Hernandez from Carleton College, who's not here with us today. I'd also like, and we can hold our applause for these two, but Tina Chang. And also Abe Walker. Our instructors are amazing people who really help the scholars learn how to make observations in nature, ask questions, help them understand what were the different field methods they should be using, their analyses, and really were the ones who were the core of really helping with this learning the field methods and how to conduct research in the field this summer. So I really want to take a big shout out, give a big shout out to you all for helping out. So I'm going to call out the names, and then after I call out the names. So the next people I want to give thanks to are Nick Macias, who can't be with us today, Melissa Cronin, and if the next two people can stand, Kyle Reed, and Vicky Tang. These were some of our mentors this summer who really were our mentors really help, talk to the scholars a lot about, well, one, they help in the field with collecting data, helping us get around, but they also really help with a lot of the DEI trainings that we have, diversity, equity, inclusion. We do a lot of debriefs, which everybody, that's probably a favorite word, our different experiences and how we feel when we're out in some of these places, especially where some of us might not feel like we're reflected. And so really want to thank them for being on board with us and all the hard work that they do as well. And then finally, there's two people who I think that without them, this program wouldn't be able to really function. These two people are our logistics staff and should not go without thanking. They are responsible for making sure that we know where we're going when we're going from place to place, making sure everybody's got enough food. When anybody has a question, they usually go to them. And really, this program wouldn't, and also checking in and out here, but it really wouldn't function because it's kind of like the day-to-day nuts and bolts of what make these things work. And so I want to have Alex Bevan and Jessica Weichert stand so we can. All right, so with that, thank you all for coming. And there's some snacks out in the lobby. I really encourage the scholars, the scholars will be here, and I really encourage, we can't go anywhere without the bands. But I really encourage our guests who all came today, please engage with the scholars if you want to learn more about their experiences over the summer. Before we go, I also want to thank a few more people. I want to thank the EEB department for hosting the Doors to Conservation Scholars Program, UCSC. I also want to thank Alex Jones, Miranda Allen, who's here with us, Nancy Heichman, and Linda Scholl. They were some of the folks on campus that helped a lot with our professional development. So I want to make sure that we give them a round of applause too. And with that, thank you all. Oh, wait, there's one last one.
I'm going to add one more. I just want to introduce you to Justin Sirachi. So some of our interns who were in Santa Cruz this summer, so summer 2, was we talked about the internship program. Our interns came and presented to the new cohort what they're going to be doing next summer. Okay. And Justin was interns who worked with Soledad on the project. But those of you who are really excited about big animal studies, please talk to Justin.
right, thank you all.
Anybody else wants to recruit a 10th grade for next summer for 2020?
Yeah.